Yes. So far in our course, we have finished a big part of our syllabus. We have 2 units left. That's translation and then regulation. So we learned about DNA, the structure, and we learned about its structure. Then we learned about the replication of it. Then we learned about transcription, and we also learned about change, right? What do I mean by change? Change in the genome can happen because of mutations, and it can also happen because of recombination. In mutations, it could be spontaneous mutations. They are because of replication errors and so on. There are also others called as induced mutations. Induced mutations could be because of chemical mutagens or physical mutagens. In recombination, the change in the genome or the genetic information can also occur because of recombination. In recombination, we learned about two types. One is about homologous recombination, and the other one is site specific recombination. There are also some other things in the chapter on site specific recombination. We learned something about transposition as well, right? And in replication, we learned eukaryotic replication and also prokaryotic replication. Most of the focus was on prokaryotic replication. Then we learned about transcription, and we learned about the enzyme that is required, that is RNA polymerase. In eukaryotes, you have 3 different RNA polymerases, of which RNA polymerase 2 is the one that actually transcribed most of the mRNAs. And the substrates are NTPs, right? Nucleotide triphosphates or ribonucleotide triphosphates. And in eukaryotes, we also learned that the genes contain introns, intervening sequences. So we had a chapter on splicing. In splicing, we learned about 3 classes: spliceosome mediated one, class 2 introns, class 1 introns. We also, in the same chapter, also learned other modifications, post-transcriptional modifications, that is RNA editing, right? And that splicing is about joining of exons. This majority of it is in relation to eukaryotic genes. And one more important question I actually told you in at level of transcription or upon splicing is about the differences between prokaryotic and eukaryotic mRNAs. There are certain differences that is also representative of how the genes are present on the genome. And from now on, what we should be focusing is on an important chapter, translation. And after that, we will have, that's the end of it. Of course, if you consider the central dogma wise, you have the DNA and transcription, splicing, you can say, and then translation. Translation should give rise to proteins. And this until from DNA to that of splicing or say transcription, we are dealing it in the language of nucleotides or nucleic acids. But now we are dealing from here on, we will deal in the language of amino acids. Right? There are 20 different amino acids, and they all are unique in their side chains. What is the common group? In the amino acids, you have an amino group. Okay, something is wrong, but okay. NH2, say, I should have put N here, but yeah. C, COH, that's the common general formula for an amino acid. So all the different amino acids, they differ in the R group, the chain. And there is proline, which has a difference also, amino acid. So from here on, we are talking in terms of in amino acids. And the important thing about translation or learning is able to appreciate the beauty of how things have evolved to translate the genetic information that is present in the language of nucleic acids or nucleotides to that of amino acids. And upon translation, we will get proteins, and proteins can have varied number of amino acids and type of amino acids. That means you can actually generate very complex or diverse complex structures, and each of them may have some function because of their structures. When you have several amino acids form, I mean, as a protein, then they can fold up upon themselves based on the interactions of the side groups, and then they might be able to interact with some other molecule. All of biology is about interactions. There is no protein that doesn't interact with anything. At least it will interact with some other another protein of its same kind, like something like homodimer, for example. So everything in biology that you see as a dynamic entity, for example, a cell, it's about interactions. If you think of biology in one way, you have information, that is the genetic information. That genetic information will be exhibited as an interaction. And that interaction results in a consequence or an effect, right? It could be an enzyme reaction, it could be a transporter, it could be a modifying enzyme, something like that, right? So genetic information is how it translates into an action or an effect. Is information, genetic information gives rise to molecules that can interact. And that interaction, when you have A interacting with something else, something like B, it might give rise to C, right? This is the consequence of it. So it's a way of seeing how biology works. And that is amazing when you break down things into bits. Often what we will see is, yes, directly upon transcription and translation, a protein is formed, protein is an enzyme, and that's it. Instead, if we talk about interactions, then you will find the beauty of evolution itself. Why I am emphasizing on interaction, excuse me. Okay, why I'm emphasizing on interactions is that the interactions could be fruitful, could be useful, may not be useful, or may not be any functional, right? And here selection happens. Those interactions that give that produce favorable consequences will be selected, and the rest will be eliminated. So if there is a genetic information, a gene has newly formed, if it did not produce a protein that can interact with something, then eventually that will be eliminated. If there is a gene formed and it allows the formation or interaction of formation of an interacting molecule with something else, and the consequence, if there is no consequence to the interaction, then also those genes will not be selected. But if the consequence is good for the cell, that means it's favorable, that cell is benefited, and then those with that particular gene will be selected. You don't have to worry about it, but it is how you build an image of biology and a thought process. There is the biological way of thinking. You have a mathematical way of thinking, and so on. Some way, when you see biology, you have to think between information and consequence, a physical consequence. Okay, so as such, you can think of the information is kind of abstract. Assume it's kind of abstract, and information that is non-materialistic can become materialistic and as a physical entity. And that's the beauty of how you see biology. Okay, so what I'm going to do in today's class is just discuss a few things and talk about open reading frame, genetic code, and so on, so that it will be useful for the chapter on translation. And I'm also taking this because I thought it will be useful for the assignment, the compulsory assignment that you are doing for 4 marks. Right. So replication, you have DNA. This is eukaryotic gene. There are introns. Those are the intervening sequences, and there are this is usually present in the nucleus, always present in the nucleus. The In eukaryotes, either DNA is also present in mitochondria, and in plants also in chloroplasts that have origins of bacterial in origin. But anyway, the otherwise majority of the DNA or all of the DNA is in the nucleus. And then transcription gives rise to pre-mRNA. That is, it still has the introns, only that now it is in the form of RNA. And upon splicing, that is joining of exons, you will get the eliminate introns and fuse the exons in continuous way. And now we have a start codon here, and here we'll have a stop codon. So as we'll see, as we may see in the future, there is also a process called reverse transcription, that is in retroviruses. We have actually seen it in retro transposons, where an RNA intermediate is converted into DNA, or it's a DNA form of an RNA intermediate is made by polymerization, and that is by the process is called as reverse transcription, and the enzyme is called as reverse transcriptase. And this also happens in the nucleus. And then the mRNA is transported out of the nucleus in the cytoplasm. Either it is bound to endoplasmic reticulum, that is the rough endoplasmic reticulum, or freely in the cytoplasm. It depends on the product of the mRNA. You will have ribosomes binding and translating the mRNA to form proteins. Okay, so we are going to study about this. In that, when we study it, we are supposed to study the mRNA of it. Then we are going to see about the ribosomes. Then we also have to read about tRNAs. These are the components required for translation to happen. If you have tRNAs, it's actually charging of tRNAs. So we'll have to study about tRNA synthetases and so on. And then we have something as a mechanism we need to study, that each is about simple, like initiation, elongation, and termination. Again, as usual with every chapter that we are studying, we have to study it in how it happens in prokaryotes and how it happens in eukaryotes, the similarities and differences. Okay, so what we'll see is a few basic details about genetic code. Genetic code, you have to read what is a codon to start with. A codon is a triplet. Triplet means three, and triplet of three bases. Right? You all know that you have studied it. So when they were, we will discuss probably about how they have found it out at a later class, in later classes maybe. But otherwise, if there was how DNA can, or nucleic acids can be, the information that is in the form of nucleic acids can be studied as, or they converted to information in terms of amino acids. We have four bases and 20 amino acids. Right? How can four bases be done? So if you take two, if you are starting to read, if you start to read as one base codes for one amino acids, then we only have only four of the bases could be coded. Right? If we had, if you take two bases at a time, I think you can form a combination of eight or so, I think. 4 into 4, probably 16. So you'll be able to read about 16 amino acids. But if you take about three bases at a time, then you will generate about, you can read about 64 amino acids. I mean, that's not, you only have 20 amino acids. But in that case, if you are starting to read bases in steps of three, then you will generate about 64, each one specifying for one amino acid wise. If you think of it that way, then it is about 64 amino acids, but we only have 20. So as the experiments were done more and more, what they found was many codons, that is genetic code is degenerate. What means one codon codes for one amino acid. That is possible. More than one codon can code for one amino acid. Okay. The one difference, if I have to make it as a statement, is nowhere you will find one codon coding for, this will not happen, coding for more than one amino acid. One codon will only code for one amino acid. Many codons may code for one amino acid, but you will never find one codon coding for more than one amino acid. There are a few exceptions. Again, if you study biology, it is said something like this: biology is the science of exceptions, meaning to say that you have rules and there will be some exceptions to those rules. You can find many of them, many examples for that. It's a science of exceptions in a way. Okay. I mean, it mostly depends on the context and how you define things. So this is the first codon, first position, this is second position, and this is third position. So I'll try to redo this. So you have, I will try to take this one here. First codon should be taken here, so I would take as U. Second position on this axis, x-axis, U, and this is the third position. So I have to take U, and that is U, U, U. Say I want to do this. The first position I am taking is from C, right? You can actually fill it up, C, and then you have A based on this, and on the third position again you have A here. That's how you actually can make a table by yourself. I don't need that you mug up this. You don't have to do it at all. All you have to remember is start codon and stop codon. Start codon, and these are the stop codons. You should have that in your mind all the time. So AUG, because we are doing it in RNA, and UAA, UAG, UGA. These are the stop codons, and AUG is the start codon. So there are certain rules about open reading frame. That is, you will have to read it in continuity. That is ORF, open reading frame. That is, it has to start with a start codon. That means it should invariably have AUG, and then you should have continuous codons, and then it should end up with a stop codon, say for example UGA. Each of these codons is made up of 3 bases, right? That is a codon, and there are no commas or spaces in between. As I said, biology is an science of exceptions. You can think of it. There is, in some cases, in different organisms, there might be a little bit of difference. In some cases, GUG is also counted as start codons. But predominantly in prokaryotes as well as in eukaryotes, AUG is the start codon. And I said that the codons should be read continuously without any spaces. But then again, there is something called frame shifting. There is something called as frame shifting. Ribosomes sometimes can move +1 or +2 or -1 or -2. It can jump a base and start reading, or it can move back a base and start reading again. So there are exceptions, but generally speaking, an open reading frame is continuous. It has start codon, then it has several codons that are continuous, and then ends with a stop codon. If we get time and we are talking about the coronavirus, then we will talk about these frame shifting as well. Those are some of the interesting things that you can see. The objective of this class is to help you with the assignment as well. I gave you some sequences, and then I asked you to look into the sequences and identify certain things and so on, compare them. In one of the questions, at least, is your ability to identify an open reading frame. Here is a sequence that is given in double-stranded, and that double-stranded one, excuse me, let me see. Yes, there is a double-stranded, and if you are looking at the sequences, you can start looking at if there is an ATG, that is the start codon. Here we are dealing it at the level of DNA, so ATG. And you should be able to read continuously in steps of 3, and then you got a stop codon. Right, that is a stop codon. So you have an open reading frame. That is what is indicated here, ORF1. Then it is not the end. If you find one open reading frame, and that you are, if you don't look further, then you are actually not doing justice to the assignment. You are supposed to look further beyond what is there, and then have a look at further ones. That is, the second open reading frame starts from here, ATG, and then you will have this something like that, and TAA, you got a stop codon. So if you say, for example, so let me finish this. So this is open reading frame. Say, for example, you found an open reading frame, and there is, say this is TAC, and you don't have a stop codon, then it is not an open reading frame. The rules of open reading frame is that it should have a start codon, continuous codons, and a stop codon, all in one frame. What is a reading frame? Again, you can start reading from here. That is, if I'm reading frame 1, and then I can keep going it. This is reading frame 1. If I am trying to read in frame 2, then I would start here, 3 bases at a time. Just I started at a different position like this. If I were to start again in a different location, then I could start in this location, that is 3, all in steps of 3. right? The first one is, this is one reading frame, second reading frame, third reading third reading frame. Reading always should happen from 5 prime to 3 prime. There is no exception to this. You should not read it this way. But there is also a 5 prime on the complementary strand. You see 5 prime to 3 prime. So there is a possibility that open reading frame could be on this. Again, if you're counting in terms of reading frames, you can start reading from here and see if there is anything or not. Here you find ATG, right? I'm reading it in a different ATG. Then you have continuous reading. You can go GAT, CTG, ATG, AGT, and so on. I can keep going. GAT, GAT, GAC, ATG, and TAG. TAG is a stop codon. So if I start, this is one reading frame. The second reading frame could start from here, reading in steps of 3. And the third reading frame could have started here, right? So any sequence, any double-stranded sequence that is given to you, or you convert it into double strands, you will have a total of 6 reading frames. Not that all of them should have or would have an open reading frame, but the reading is where do you start, right? So 3 from this side, 3 from for the top strand, 5 prime to 3 prime direction, 3 reading frames for the bottom strand from the 5 prime to 3 prime reading frame. Okay, this is something in my research I come across this a lot. These are called as overlapping reading frames. You have one reading frame from here to here, right? And another reading frame in a different frame, it is from here to here. So that's a good organization. And in the same DNA, you're now having another open reading frame, that is ORF3 here. I hope you understood. When you are doing the assignment, you are supposed to convert the sequences into double-strand DNA and then look through carefully. The more you do this exercise, the more closer you will get to the way of thinking in terms of information, or actually bioinformation. And that will take you a long way. So, and also we have discussed something else, is about gene density. It is to say how many genes are there, number of genes present in 1 kb of DNA. That's approximately. But the point is, if you give a small fragment of DNA, how many open reading frames are there in it? It's very typical of viruses that they have a lot of overlapping reading frames. If you go back to this this slide, you can think of this as one. It is one open reading frame. Always the arrowhead indicates the 5 prime to 3 prime, right? Arrowhead is towards the 3 prime, the direction in which you're reading it. You can also, this is also another open reading frame, right? These are overlapped open reading frames. And for the bottom one, you should put it something like this. That's the direction of reading. Okay. So when we are representing, we do put something like this, ORF1, 2, and 3. In viruses, you find high gene density. That means within a small fragment, you might have more number of genes encoded, or they express. When they express, could produce proteins or functional RNA. Next, we have bacteria. And in bacteria, you will have operons. What are operons? Operons are a collection of genes, usually connected in function as well. That means they produce enzymes of a pathway, or they have related functions. The operon is something like this. It has one promoter. This is +1. And during transcription, will produce this whole mRNA, a single unit. and upon when there is a terminator, then it will stop there. And this is one whole mRNA, and it has three independent open reading frames, and each of them would produce one protein. Right? That is an operon. It's very typical of prokaryotic genes or genomes to have organized their genes in operons. It's easy um, to regulate them. Say, for example, I don't have lactose, then it's smart and logical to be able to suppress all the genes that are involved in lactose metabolism, to suppress in one go instead of having to suppress each one, right? So it makes sense for bacterial or prokaryotic type of lifestyle to have organized their genes in as bits or as operons. And that is where we also, when we are discussing about the mRNA of prokaryotes, we said they are polycistronic. They encode one mRNA has multiple open reading frames like this. So they are polycistronic. In eukaryotes, they have genes which should be, and they are usually monocistronic, and they have, as we discussed just a little while ago, they have, upon splicing, they will have one open reading frame. That's very typical of eukaryotic genes. Okay. So there is something that I want you to address this. I will stop in 10 minutes. I have another meeting to start. So I have a code here. We will do it as per what I have discussed about the rules of codons. Okay, assume that this is open reading frame. You have to read in triplets or three letters. So here I have PLB, int, he, something, something, something. I cannot pronounce them. And I gave, we remember mutations, right? In mutations, we read about what to do. We read, it is, I mean, we had insertions, deletions, rearrangement, inversions, and so on. So the same thing I would want you to do, except that I've just made this theoretical kind of one to understand what I wanted to explain in the next slides. I hope you, okay, maybe not. So in this key here indicates that at third position, add a Z. Okay, so let's write this PL, and this is the third position. So I have to write a Z. And then I have to move this whole, from here on, I have to move it this way. And I have to still read it only in three letters. So next one I have to write is bin. And next would be the fly. The fly. And AND. And then get the mop. Right, I hope you can keep on reading this. Let me finish it anyway because we started it. Mop, and then hug mom. Hug mom. And I forgot to do this. This is the second key that is also there. What did I do? So I have a second part to do it, second modification to do. That is going to, let me check, at 31 position. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That is 30. And in this place, I am supposed to add GA here. Okay. Hug mom, and then wait a second. 1 second. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If I add this, what will it become? GAA. Sorry, it is minus.
So you have to remove GA.
That's right. Thanks for this. Yeah, it is minus GA. So I made a mistake there. Yes, thank you. So I had to remove this GA. That's the key there. So what I have to do is, if I remove it, then it's much easy, simple. And so these two are gone. And now I have a and buy her car. So this is just to illustrate how a reading frame changes, right? The sentence, if you are reading the sentence as it is, it has no sense that we can actually make of. If you're reading in basis of three or letters of three, but if we had something inserted and something deleted, then we have some meaning to it. That's way it is some that is how an open reading frame also changes. But anyway, there is a message here also that all moms work a lot. Please bin the fly, remove the bad things, and get the mop to clean up. Then this is more important if you are not doing in the kitchen. Hug mom, and you should be able to tell them how much work they do, because it's underappreciated in all over the cultures in a way, because they don't nobody pays for doing the housework. But I think every husband's every working person's half the salary belongs to the wife of the house, because she is the one getting the house running, and it should be appreciated. And I hope you will all be able to buy her a car or something that you would appreciate. It's just for fun, but the point, yeah, you should take it also as seriously maybe. The point is how insertions or deletions could change the meaning or the information, is what we are actually doing here. That's exactly how the mutations also happen. We did not, I think, explicitly discuss something about nonsense mutations or frame shift mutations and silent mutations. If this is a reading frame, this is the last slide. I'll stop in 5 minutes because I need to go. So this is a reading frame, and each of them is coding for one amino acid like this. This one you can remember, ATG codes for methionine, and this is stop codon here. And if we have a change here in this C, and that C is replaced with A, can you recollect if it is transition or transversion? A pyrimidine is replaced with purine, so it is transversion, right? That is adenine. And so now TCG is a sense codon because it was coding for serine, but now it is coding for TAG. It's a stop codon. Those mutations that that produce a stop codon within a reading frame are called as nonsense mutations. Those codons, some of the codons do not have any change at all. I think this is the place. Here you have AGC. The C is changed. It's a transition because a pyrimidine is replaced with a pyrimidine. Substitution, and it is a transition. And AGC codes for serine, and AGT also codes for serine. There is no difference. And those mutations are called as silent mutations. And there is the frame shift mutations, like I was describing here, where you can have deletions or insertions which are not multiples of 3, which result in the change in the reading frame, and because of which a meaningful sentence might become meaningless, or a meaningless part could become a meaningful one, or there will be a serious alteration just because you inserted one base here. All the all these things have changed, right? Because you're reading in multiples. That's an example or an analogy for frame shift mutations. Okay, so in frame shift mutations, what did we do here? Here is a G is added, because of which the old G here is moved here. This is the one. So serine. So this is serine, and so is this. But the next G in this position, it was serine actually, but now the reading is as GAG. That's glutamic acid, and CTA for leucine, and then you encountered a stop codon. Otherwise, the stop codon in the original sequence, let me erase it and try. So this is a TAG, but it is not in one codon. Here it is part of this TAT, and AGC is another codon. But because there is there is an insertion of an additional G, then now the T that was present here has moved here, and now you found it formed a stop codon, and that is where then there is um, premature termination of translation. And also you should notice that the whole amino acid sequence also changes, right? And I will just stop with one point, is that, say for example, instead of one G here, I added GAC. Say that was happening in this location. Say if I added a GAC, what will the codon be? ATG, CAG, TCG, and then I said I'm adding GAC, and then AGC, sorry, AGC, TAT, and AGC. Is there a shift in frames? This is the new codon that has been added. This was coding for methionine, CAG for GLN glutamine, TCG for serine, GAC would code for some other amino acid. And now again you have AGC that's coding for serine, TAT, tyrosine. The reading frame has not changed. That's why when we are defining about frame shift mutations, you should say when there is a deletion or insertion of bases which are not in multiples of 3. Okay. In this situation, is it a mutation? Yes, it is a mutation. But is it a frame shift mutation? It is not a frame shift mutation. It is just an insertion mutation. One extra amino acid is present within the coding or within the protein, the product that would form. Okay. I will stop here. Let me stop recording.